When you hear the words forensic investigation, your mind probably goes straight to, like, a high-tech CSI episode.
Right, yeah. Yellow police tape, fingerprint dust.
Exactly, the dramatic reveal of a suspect's identity.
Yeah.
But there is this microscopic stakeout happening right now that is, it's arguably far more complex.
Oh, absolutely.
And it is happening inside your own body. It's in the food sitting in your fridge and, even in your city's wastewater.
Yeah, it's literally everywhere.
Today, we're doing a deep dive into the science of identifying microbes with PCR. We've got a fantastic stack of research on microbiology here. And the mission today is to figure out how science shifted from trying to coax microbes to grow in a little glass petri dish to reading their DNA like a barcode.
Which is a shift that completely changed our definition of what a microbial species even is, honestly.
Totally. Okay, let's unpack this. I feel like we have to start with what PCR identification actually means at its core, because we've moved way past just staring at bugs under a microscope, right?
Oh, yeah. We have entirely bypassed the microscope. Identifying microbes with PCR, which stands for polymerase chain reaction. By the way, it means detecting and classifying organisms like bacteria or viruses by reading the unique signatures in their genetic material.
So not just looking at their shape or whatever.
Right. Historically, microbiology relied on, coaxing a bacterium to multiply on an agar plate until it formed a visible colony. But with PCR, we use a lab technique that targets a specific microscopic stretch of its DNA and just copies it billions of times.
Until you have a pile large enough to actually read.
Exactly.
So it's essentially a molecular photocopier.
That's a perfect way to put it, yeah.
But before we get into the nuts and bolts of, like, how this molecular machinery works, we really have to look at where it came from. Because the invention of PCR is a tale of two incredibly different environments.
It really is.
We're talking about a late-night drive in California and a boiling pool of water in Wyoming.
Yeah, it sounds like the setup for a sci-fi movie, honestly. The initial spark, the photocopier concept itself, was dreamed up by a biochemist named Carey Mullis.
Right.
The origin story in our sources traces back to April 1983. Mullis was on a nighttime drive along California's Highway 128. He was working for the Cetus Corporation at the time.
Just driving through the dark.
Yeah, and during that drive, the conceptual geometry of PCR just clicked for him.
Right.
He realized that if you had two short synthetic pieces of DNA, which we call primers,
Good primers.
right, and they flank a target sequence, if you have those, plus an enzyme designed to copy DNA, you could just repeatedly heat and cool the sample to trigger a chain reaction.
Heat it up to separate the double helix, cool it down to let the enzyme copy the strands, and just repeat that loop.
Exactly. And he actually won the Nobel Prize in Chemistry in 1993 for that late-night revelation.
Wow. But, having the idea on a dark highway is very different from actually making it work in a physical lab.
Oh, huge difference.
Because there was a massive physical roadblock to his brilliant idea, which brings us to the boiling pool of water.
Yes. That roadblock was the heat itself. See, the near boiling temperature is required to physically separate double-stranded DNA. It will completely destroy ordinary DNA copying enzymes.
Because they're just proteins.
Exactly. It is exactly like boiling an egg. The heat permanently tangles and denatures the proteins. If you used a normal polymerase enzyme from like a standard lab bacterium, it would literally cook and fall apart in the very first heating cycle.
So what did they do early on?
Well, early on, scientists had to physically open the tube and drop in fresh enzyme after every single heating step.
Oh my God.
Yeah, it was agonizingly tedious and incredibly expensive.
Which is where Yellowstone National Park enters the picture to completely save the day.
Right. So in 1966, this microbiologist named Thomas Brock was studying the hot springs in Yellowstone. And he discovered a bacterium thriving in near boiling water around like 70 to 80 degrees Celsius.
Just living its best life in boiling water.
Exactly. He named it Thermus aquaticus. And because this organism spent millions of years evolving in a scalding environment, its DNA copying enzyme, which we now call Taq polymerase, is completely heat stable.
It just shrugs off the heat.
It literally shrugs off the intense heat of a PCR machine.
I have to pause and marvel at the profound irony of this. It's like building the ultimate metal detector, but you can only build it by extracting iron from the very soil you're trying to scan.
Oh, that's good, yeah.
The tool that is now used to identify countless microscopic life forms was literally mined from the extreme biology of one.
Yeah, we leveraged the survival mechanism of an extremophile to unlock the biology of, well, everything else on the planet.
It's wild. So, okay, we have this heat-proof engine from the boiling springs.
Right.
But how does it actually drive the process of making the invisible visible? Let's walk through the actual mechanics of making a billion copies.
Okay, so the entire reaction happens inside a machine called a thermal cycler, and it's this beautifully orchestrated three-step thermal dance.
Thermal dance? I like that.
Step 1 is denaturation. The machine heats the liquid sample to about 94 to 98 degrees Celsius.
So basically boiling again.
Right, and this intense thermal energy physically overcomes the hydrogen bonds holding the DNA double helix together, so it acts like a broken zipper, essentially, splitting the helix into two separate single strands.
Got it. So we've unzipped the DNA with heat.
Yep, and step 2 is where things need to cool down a bit. This is called annealing.
Annealing, okay.
The machine drops the temperature to roughly 50 to 65 degrees Celsius. And this is where those synthetic primers that Mullis dreamed up come into play.
What has he thought of on the drive?
Exactly. They're short stretches of DNA, usually about 18 to 25 bases long, explicitly designed by scientists to perfectly match the sequences that flank the target gene you want to study.
Like framing the target.
Right. And at this cooler temperature, they bind, or anneal, to the single strands of target DNA.
So they basically act as molecular bookmarks, dropping onto the unzipped strands to tell the enzyme, hey, start your copying process right here.
Precisely.
Yeah.
Which brings us to the final step. We've broken the zipper apart with heat. We've laid down our synthetic bookmarks at a cooler temperature. The only thing left is for that heat-proof engine to actually build the new DNA strand.
Right, the Taq polymerase.
Step 3 is extension. The thermal cycler warms back up to about 72 degrees Celsius, which just happens to be the absolute optimal cruising temperature for our hero from Yellowstone.
Taq polymerase.
Exactly. The enzyme grabs onto those primer bookmarks and rapidly builds out the new complementary strands of DNA by pulling free nucleotides from the surrounding liquid.
Wow.
So you started the cycle with one piece of DNA and you end the cycle with two.
And then the machine just aggressively repeats that loop.
Over and over.
Unzip with heat, drop the bookmarks, build the strand, and because it doubles every single time, the math scales unimaginably fast.
Oh, it's exponential.
Yeah, if you run that cycle 20 times, your single starting molecule turns into roughly 1 million copies. At 30 cycles, you cross the billion copy mark.
It's staggering.
But wait, so if it's exponential, why do we ever stop cycling? Doesn't more cycles just mean a more sensitive test with absolutely no downside? I mean, if I want to be incredibly certain a specific microbe is hiding in my sample, shouldn't I just run the machine for like 60 cycles to amplify it as much as possible?
See, that assumption makes intuitive sense, but it hits a biological wall pretty fast. Because the technique is so exquisitely sensitive, overcycling is actually one of the most dangerous pitfalls in a laboratory.
Wait, really? Why?
Past a certain threshold, usually around 35 to 40 cycles, the Taq polymerase starts getting a little sloppy.
Mm-hmm.
Yeah, the primers might start binding to non-specific areas of DNA that sort of look like the target but actually aren't. Or worse, the machine starts amplifying microscopic background contamination from the lab environment itself.
Ah, okay, so it's like turning the volume on an amplifier all the way up to hear a really faint whisper in an audio recording.
Yes.
Eventually, you lose the whisper entirely, and you just get deafening, distorted static.
That is a perfect visualization. You start manufacturing false positives out of thin air. We'll explore that contamination danger more deeply in a moment, but first, we need to clarify a foundational rule of this technology.
Okay, what's the rule?
Copying DNA tells you absolutely nothing about the identity of the microbe. It just hands you a massive, unreadable pile of genetic material.
Right. Having 1 billion copies of a single page doesn't help you at all if you don't know what book it was ripped from.
Exactly. The true genius of modern microbiology was deciding which specific piece of DNA to copy in the first place.
And to understand how we chose that piece of DNA, we have to look at the work of Carl Woese.
Yes, back in 1977, which is years before PCR even existed.
Right.
Woese was at the University of Illinois relying on this grueling, agonizingly slow technique called RNA fingerprinting. He was comparing the sequences of ribosomal RNA across different organisms, and he hypothesized that these ribosomal genes act as a sort of stable molecular clock.
A molecular clock.
Yeah, because every living organism requires ribosomes to manufacture proteins, right? Meaning the genes that build them mutate at a very slow, highly predictable rate across evolutionary history.
So they don't change much.
Barely at all.
And he used that molecular clock to completely redraw the map of biological life. Like by looking at these slow mutating sequences, Woes discovered that some microscopic life forms we had previously classified as weird bacteria.
Right.
They were actually so genetically distinct that they formed an entirely new, separate domain of life, which we now call the archaea.
Yeah, doing that without the massive amplification power of PCR is just staggering to think about today. It was a monumental effort.
Incredible.
But when PCR finally hit the mainstream, scientists quickly merged Mullis's amplification photocopier with Wose's molecular clock. And the result was the targeting of the 16S ribosomal RNA gene.
The 16S gene.
Yes. This is a stretch of DNA, roughly 1,500 base pairs long, present in every bacterial cell in existence. Because it's universally required for survival, but it still contains slight regional variations between different species.
So it's sort of the gold standard for identifying who is who in the microbial world.
Exactly.
Like the 16S gene acts as a universal UPC barcode on grocery store items. Without this barcode, we were essentially walking through a massive supermarket with all the lights turned off, thinking the only food that existed was the 1% we managed to trip over in the dark.
What's fascinating here is how the barcode completely dismantled and rebuilt the foundation of the science.
Because of the plate count anomaly.
Yes, the great plate count anomaly.
Yeah.
Staley and Konatka coined that term in 1985. Basically, if you take a single drop of seawater or a pinch of soil and look under a microscope, you see thousands of microbes. But try to grow that exact sample on an agar plate with lab nutrients. Almost nothing grows.
Over 99% of environmental bacteria just flat out refuse to grow in a lab.
Exactly. And previously, identifying a microbe mandated that you culture it. You relied on, 19th century frameworks like Hogus postulates, where you had to isolate and grow a pure culture to prove it caused a disease.
Right.
Or you used the Gram stain from 1884, washing bacteria in purple and pink dyes to look at their cell walls. It was rigidly a culture-first science, but the 16S barcode dragged microbiology into a sequence-first era.
It's a total paradigm shift.
Today, culturing is increasingly the exception. You only really do it when you specifically need the live organism, like testing if a colony is resistant to a new antibiotic.
So once that amplification photocopier met the universal barcode, it unleashed a toolkit that completely rewired public health, forensics, and our understanding of our own bodies. Let's trace how this plays out in the real world.
Yeah, let's do it.
Starting with diagnostic PCR, I mean, you listening have probably had a swab shoved up your nose recently, which means you've directly interacted with this technology.
Oh, absolutely. During the COVID-19 pandemic, diagnostic PCR became a household term. Now, SARS-CoV-2 is an RNA virus, meaning its genetic code is in DNA. So labs rely on a variation called RT-PCR, or reverse transcription PCR.
Okay, how does that work?
First, an enzyme reverse engineers the virus's RNA into DNA. Then, the thermal cycler uses primers explicitly designed to seek out and match viral genes, like the blueprint for the spike protein, and begins the amplification cycles.
And the pandemic also introduced the public to the seed value, right? The cycle threshold.
Yes. This is a direct measurement of how hard the machine had to work to find the virus. The thermal cycler uses lasers to measure a fluorescent signal that grows brighter as more DNA is copied.
Okay.
The C value is the exact cycle number where that fluorescence crosses a detectable threshold. So a low C value, like 15 to 20, means there was a massive amount of starting viral material in the patient's sample. It crossed the finish line very quickly.
Got it. And a high one.
A high seat value, like 35 to 40, means there was only a microscopic trace amount, requiring dozens of cycles to finally detect. That specific application, run hundreds of millions of times globally, stands as the most consequential real-world deployment of PCR in human history.
The stakes are just incredibly high. And not just for viral pandemics. The same genetic detective work is used in high-level forensics. Our sources detail the FBI investigation into the 2001 anthrax letters here in the U.S.
Yeah, that's a fascinating case.
Deadly spores were sent through the mail, and investigators needed to know exactly which lab they originated from.
Right, and scientists from the FBI and U.S. ARI turned to PCR-based genetic fingerprinting. By amplifying specific genetic markers, they identified the spores as the Ames strain.
But they went deeper than that, right?
They did. They compared minor genetic mutations against samples collected from various biodefense labs around the world. Through PCR fingerprinting, they traced the weaponized anthrax back to one specific container, a flask designated RMR1H29, sitting in a lab at Fort Detrick.
That is literal microscopic forensics. And we see that same precision in food safety. If you think about the vegetables in your crisper drawer, PCR plays a massive role in keeping them safe.
It does. Our notes highlight a massive E. coli outbreak in Germany back in 2011.
The E. coli 0104.H4 outbreak.
Yes. It was highly lethal, moved incredibly fast. Early on, authorities strongly suspected imported Spanish cucumbers were the source.
Which triggered massive import bans.
Exactly, causing devastating economic damage to farmers in Spain. However, by deploying PCR-based typing alongside traditional epidemiology, investigators mapped the exact genetic signature of the outbreak strain.
And what did they find?
They traced it back to its true source, a batch of contaminated fenugreek sprouts. The PCR data fully exonerated the cucumbers and pinpointed the real threat.
Here's where it gets really interesting, though. Whether it's saving hours, diagnosing sepsis in a hospital, or tracking a global outbreak in a city's wastewater, this invisible barcode reading is like the unseen load-bearing wall of modern society. And it's even redefined how we view ourselves as a species.
You're talking about the Human Microbiome Project.
Exactly, launched in 2007.
That project leaned heavily on 16S sequencing to explore the ecosystem inside you. It was built on the pioneering work of microbiologist Norman Pace in the 1980s. He was one of the first scientists to realize you could extract DNA directly from environmental samples without ever trying to grow colonies first.
Which bypassed that whole 99% blind spot.
Exactly. By sequencing the barcodes found in human samples, scientists cataloged thousands of unique bacterial species living in our guts, on our skin, in our mouths, the vast majority of which had never been grown in a lab.
It proved unequivocally that humans are just walking, talking ecosystems, and PCR provided the first accurate map of our microscopic passengers.
It really did.
But, any tool this powerful has an Achilles heel. Because PCR can amplify a single molecule, it is exquisitely vulnerable to, well, lying to us.
Yeah, the sheer amplifying power of PCR is absolutely its greatest vulnerability. If a stray fleck of skin from a lab technician, a contaminated chemical reagent, or even degraded DNA floating in supposedly pure lab water makes its way into the tube,
The thermal cycle will just dutifully amplify that mistake a billion times over.
a billion times.
And suddenly you publish a paper claiming you've discovered something extraordinary, but it's actually just a fleck of modern dust.
This specific vulnerability has burned the ancient DNA community repeatedly.
Right. There's that famous cautionary tale from the year 2000.
Yes, involving a paper published in the journal Nature. A team of researchers claimed they had successfully revived 250-million-year-old bacterial spores trapped inside ancient salt crystals deep underground.
A quarter of a billion years old.
Yeah.
That predates the first dinosaurs.
It was a sensational claim, but the wider scientific community immediately pushed back. Critics analyzed the data and pointed out that the DNA sequences of these supposedly ancient bacteria were functionally identical to modern, common contaminant species found in standard laboratories. Oh, wow. So they just amplified their own lab's background noise.
That's the overwhelming consensus today, yeah. It serves as a permanent teaching example in microbiology. Extraordinary PCR claims require extraordinary negative controls. You have to definitively prove you are not just turning up the volume on background noise.
And this vulnerability isn't just an academic debate about ancient salt. It spilled over into massive, real-world confusion during the COVID-19 pandemic.
Oh, the isolation policies.
Right, regarding whether someone was actually contagious.
Yeah, the live versus dead debate was a chaotic period for public health policy, and it stems from a fundamental limitation of the technology. PCR only detects genetic blueprints. It does not detect living, functioning viral factories.
Right, it doesn't care if the virus is alive.
Exactly. During the pandemic, patients would repeatedly test positive on an RT-PCR test for weeks or even months after their initial illness, long past the point where they posed any danger to others. Because the ultrasensitive test was just vacuuming up the degraded, shattered fragments of viral RNA that were still being cleared out by their immune system.
Precisely. At those high set values, the 35s and 40s, the machine was amplifying harmless genetic debris. But because a positive test was universally interpreted by the public and some institutions to mean actively contagious, people were barred from returning to work or school unnecessarily.
The test was never designed to tell you if the virus is intact and dangerous, just that its shattered code is present in the swamp.
Exactly.
Beyond amplifying the wrong things, there are structural limitations to the barcode itself, too. The 16S primers we use, they're called universal, but they actually have a built-in bias, right?
They do. Primer bias is a major headache in microbiome research. We call the 16S primers universal, but different evolutionary lineages of bacteria have slightly different flanking sequences.
How does that affect the test?
Imagine the primers are like a physical key fitting into a lock. If the 16S gene of one bacterium in your sample is a perfect fit for the key, the copying engine revs up instantly and efficiently. But if a different bacterium has a slightly rusty or mismatched lock, the engine struggles to get started.
And over 30 or 40 cycles of exponential copying, that tiny initial advantage just snowballs.
Yes. The bacterium with the perfect lock completely drowns out the one with the rusty lock, handing you a heavily distorted view of what is actually living in the sample.
Furthermore, while the 16S gene is fantastic at telling you the broad genus of a bacterium, say, identifying it as a staphylococcus, it often lacks the resolution to distinguish down to the highly specific strain level.
Right. Closely related bugs might have nearly identical 16S sequences, but vastly different behaviors in the human body.
Which is why we're seeing entirely new, rival technologies enter the arena. If a hospital lab has already managed to grow a culture, our sources know they're increasingly abandoning PCR for MALDI-TOF mass spectrometry.
Yeah, MALDI-TOF is incredible. Instead of reading DNA, they hit the sample with a laser, vaporize it, and measure the literal flight time of the flying proteins to get a mass signature.
It's like a rapid molecular way station.
Exactly.
And on the horizon, we have CRISPR-based diagnostics, like Surality and Detector, which hunt down specific genetic sequences without ever needing a bulky thermal cycler to heat and cool the
The
sample.
field is evolving so fast.
So synthesizing all of this, if a PCR test can amplify dead debris or fall victim to primer bias and fundamentally just tells you genetic material is present, how do doctors actually know if you, the patient, are sick?
Well, it requires recognizing that PCR is a remarkably powerful piece of a puzzle, but it is not the entire picture. Clinical context remains irreplaceable. A doctor has to evaluate the patient's physical symptoms, perhaps run a rapid antigen test that detects actively produced proteins, or occasionally use those traditional culturing methods to see if the germ is alive. A positive PCR result is a vital clue, but it requires human expertise to interpret what that clue actually means for the living, breathing person sitting on the exam table.
We have covered incredible ground today. I mean, we started at the boiling hot springs of Yellowstone, tracking how an extremophile bacterium handed us the heat-stable engine required to copy DNA.
From a bacterium that just liked hot water.
Exactly. We saw how Carl Woese's discovery of the 16S universal barcode gave that engine the perfect target to hunt for. And we mapped how combining those two breakthroughs shifted the entire field of microbiology from a slow, culture-first science to a lightning-fast sequence-first science.
Illuminating the 99% of the microbial world that classical techniques completely missed. If we connect this to the bigger picture, it highlights a crucial lesson for navigating our modern medical landscape. We are surrounded by almost magical diagnostic tools, but in a world flooded with medical data, understanding how a test works, its mechanisms, its blind spots, its limitations, is just as critical as the binary positive or negative result it spits out on a lab report.
That is so true.
Ooh.
And that brings us to a final lingering thought for you to ponder. PCR completely blew the lid off the microbial world by showing us the vast majority of bacteria we couldn't grow in a petri dish. But here is the catch. PCR fundamentally relies on universal primers.
Right.
Those synthetic bookmarks were designed by humans based on the genetic architecture of the life we already know exists. So what if our carefully designed primers are completely blind to an entirely different hidden domain of life that doesn't share that exact 16S barcode?
That is a wild thought.
What bizarre alien biology might be sitting right under our noses, swimming in our oceans, or hiding deep in our soil, totally invisible to our molecular photocopiers, just waiting for the next Kary Mullis to invent a new way to see it.
It makes you wonder what else we're missing.
Because if there's one thing this invisible biological stakeout has taught us, it is that the microscopic world is always vastly stranger and more complex than we think. Thanks for joining us on this deep dive. Till next time.